between, there's like a section here, like mine is page 66 that talks about this equilibrium, okay? And then actually in the nuts and bolts part of it under experimental, it also talks about the same equilibrium. What all of these have in common is there's something visual that you can see. So this is what we call a qualitative instead of quantitative lab. It's qualitative just because you're just going to see changes. So we have two species on either side of the equilibrium arrow. One is orange and one is yellow. So if you read kind of back and forth between those two sections, you're going to see that you're going to shock the system in a number of ways. The first thing you're going to do is you are going to add H2SO4 to your dichromate. So I guess I should go nuts and bolts first. So I'm going to go back and forth. This is page 69. It says place about 3 milliliters of dichromate. So actually under the hoods, it's from left to right. There are two chemical equilibria stuff here. These first three reagents go with your first equilibrium. These reagents go with your second equilibrium. This is your third, and this is your fourth. Okay. So the first one, it says put 3 milliliters of this yellow stuff in a test tube. So here's the deal. In front of each one of these, I have a dedicated beaker. So what I'm hoping is we can just use these dedicated beakers, okay? Because then the thing is, and I wrote this on the whiteboard, actually we've got two sizes of pipette bulbs back there by the balances. We've got these big pipette bulbs, okay? And these would suck up 3 mils, okay? So actually I'm going to go ahead and stick this one in that dedicated beaker for the dichromate. That's 3 mils, transfer it into a test tube, okay? That's how that goes, okay? Then it says add 3 molar H2SO4 dropwise. So for the dropwise ones, I have these smaller ones. So actually in the first two beakers, I'll get this done by the first two beakers, I'm going to go ahead and put this one for the 3 mils, okay, and this one for the dropwise. But my point is what we've been talking about in class. When you add dropwise, your H2SO4, check it out, you're adding H pluses. And H plus is a reactant, so you figure you should force it to form more product. Okay, so in your results and discussion, I'm going to have to see that. Actually, I shouldn't say that. You're supposed to write what you observe, right? It says record your observations when you add the H2SO4. Nuts and bolts then says, all right, we have another reagent next to your H2SO4, NaOH. The thing about NaOH, and we'll be talking more about acids and bases, is that is a base, and so that OH minus actually reacts with your H plus to form water, so that's like removing a reactant. So that little green arrow is kind of showing you would expect it to go back and form more orange, and well, let's just see if it does. Okay, so that's kind of how that goes. Right next to it is one of my favorite, your cobalt complex equilibrium. So it goes back and forth between cobalt +2 having 6 waters of hydration, it's blue, and then actually in the presence of chlorine, it will go ahead and turn pink. And it complexes with 4 chloride ions. So the nuts and bolts of this looks like this. It says place about 3 milliliters of the pink stuff, okay, in a test tube. Actually, it says not just in one test tube, it says in 3 test tubes. So you're going to run this in triplicate, basically. Okay, so this is your second equilibrium. So 1 squirt in each of 3 test tubes. It says to 1 test tube, add 2 milliliters of concentrated HCl. That's the one thing I didn't dish up yet. I have a jug back here, and just to save in time, since I'm going to do this too, these, I told you to use these for dropwise, and the cool thing about these is actually they are calibrated disposable pipettes. The top mark there is 1 mil. So I'm going to put this, actually there's one already in here. So actually when I put concentrated HCl in there, basically you're going to do 2 squirts up to 1 mil to get 2 mils of your HCl. Okay? And then check your observations. So that's what you do to one of your 3 test tubes. Okay? The other test tube, it says add about, to the second test tube, add about 1.5 grams of ammonium chloride. So the thing about, I I should say, here adding the HCl, of course HCl breaks apart into H plus and Cl minus. And the thing here is you are adding reactant. The chloride ion is one of your reactants. So you would expect it to push it that way, to the product side. That's what the green arrow is saying. When you add ammonium chloride breaks apart into ammonium plus chloride ion. Again, the chloride ion is a product. Okay, so when you add product, chloride is a reactant. Chloride is a reactant, so when you add reactant, you expect it to go that way. But it's a little complicated, because if you read in the background information, I put up in purple, actually red and purple, this equilibrium is exothermic. So the thing about, I don't know if you guys remember when we dissolved ammonium chloride, it got cold. Okay, just taking water, putting that salt in water, water gets cold. So that's very endothermic. So basically the endothermic of dissolving the ammonium chloride will remove heat, and removing heat is like removing one of the products. So according to Le Chatelier's principle, if you remove something, basically it's kind of, instead of pushing you, it's like tug of war, you're bringing it to that side. So that would favor that side, so also would adding chloride ions. So we'll kind of make your observations. So those are 3 test tubes with the cobalt chloride in it. That was 2. And then the third one you just leave alone. Okay. And then you're going to take these 3 test tubes and we're going to take them over here. I'm going to crank this up and let it boil. It was boiling a little bit ago. So we need to take those 3 test tubes and submit them in here. We might need some more test tube holders. So the test tube holders look like, well, you know what test tube holders look like. But there's extra test tube holders over here if we need a set of 3. Feel free to take pictures. This is kind of pretty, the whole pink and blue. Okay. And so the last thing is when you heat all 3 of them up, write your observations. This is where sometimes it kind of surprises me. But the fact that we're adding heat, remember, since it's exothermic, heat is a product, so adding heat should shove it this way. And let's see what we get. We might get a little bit different between the 3 scenarios. One that had the HCl, one that had the ammonium chloride, and one that didn't have anything. So write that down and include it in your results and discussion. Okay, so the third equilibrium, the stuff is under the middle hood. Nuts and bolts of this one, it says place 1 milliliter of your iron 3 nitrate in one of your cute little 1 milliliter beakers. Basically, you're going to do a dilution, okay? So find a 1 milliliter beaker, okay? So I guess when we go to get this orange stuff, we're going to use one of those calibrated pipettes. We're just going to put some of this in here and then suck up 1 milliliter and transfer it into our 100 ml beaker. Yep. And then it says go ahead and in your 100 ml beaker, add 1 milliliter of the potassium thiocyanate, which is also under there. So we'll go ahead and use the same thing. Okay, and so you just have 2 milliliters in there, record your observation, and then it says go ahead and dilute it up. After you've recorded your observation, go ahead and add about 50 milliliters of water. We could just use the gradations on the beaker to get to 50 milliliters of water. And then we're going to take about 5 milliliters of that solution, probably use a graduated cylinder, about 5 milliliters of that solution, and we're going to pour it into 3 test tubes. So finally, with our 3 test tubes, we're going to do some things after we've recorded some observations. To the first test tube, we are going to add more of the iron 3 nitrate to the first test tube. So the iron 3 nitrate, of course, has the iron 3 in it, which is a reactant, so we should expect it to go that way. To the second test tube, we're going to add the other reactant, 1 ml of the potassium thiocyanate. Again, the SCN minus, a reactant, and it should go that way. To the third one, we're going to add 1 ml of sodium hydroxide. So sodium hydroxide, you're like, well, what's that going to do? Well, the thing about the hydroxide in sodium hydroxide, it complexes with your iron 3. So actually, adding sodium hydroxide is like removing iron 3. So if we remove a reactant, it's like pulling. Okay, Le Chatelier's principle, we're stressing it. Well, how's it going to respond? It's actually going to go to the reactant side. That's what the green arrow says. So we'll record observations to each one of those. And then the last one last but not least is a solubility sort of thing. So basically silver chloride is insoluble. And so we're going to start out by nuts and bolts taking 3 milliliters of silver nitrate, okay, not silver nitrate. And then we're going to, just in that one test tube, add a few drops of, oh sorry, before I do that. So we have silver nitrate in the test tube. We're going to add a few drops of table salt, sodium chloride, okay, and in the first case it says add about 5 drops of the 0.1 molar sodium chloride. Record your observations. And then in that same test tube, we're going to go ahead and add 1 milliliter of NH3. So you're like, well, how does NH3 fit in? Well, your background information says this. Oh, sorry. Here's how NH3 fits in. NH3 actually will gobble up your Ag+. So if NH3 gobbles up your Ag+, and your silver ion is a product, then it's a pulling situation, according to Le Chatelier's principle. So you would expect basically more product to form. And what is the product? It's the dissolved species. So let's see. Okay? Look to what happens to your precipitate should go down. And then last but not least, I guess it says go ahead and do a couple doses of that NH3 solution. All right. And that's that.